So welcome back. We are going to start the second week of development. Okay, first week we have studied till implantation process, right? Till implantation we have completed from fertilization to implantation. Do you remember that? These are the competencies which are required. So let us quickly revise what has happened. Okay, once the egg is ovulated, this is the place where the sperms enter through the fallopian tube, and at the level of ampulla, the sperm crosses the three barriers and enter inside the egg and fertilize it. So we had studied what are the various layers where the sperm has to cross and what are the effects of fertilization. We have already studied that. And after fertilization, we also studied that how the cell divides through cleavage division and how it goes into the 2-cell stage, 4-cell, 8-cell stage, which is called as morula, right? Once it converts into morula, it grows into more larger in size, which is called as mature morula, and inside the zona pellucida, outside there is covering still, that is called as zona pellucida. Zona pellucida is still intact. Here the cells organize themselves into different layers. The outer covering of cells are called as trophoblast, and few cells accumulate at one corner. These cells are called as embryoblast. Okay. On 7th day, 6th to 7th day or 7th to 9th day post fertilization, the zona pellucida is removed off. Here the trophoblast starts erupting the surface of the endometrium and starts entering into the substance of the endometrium. Okay, this process is called as implantation. So this completes the first week of development. Now today we are going to study about second week of development. On 8th day onwards, what happens? Okay, so this is zona pellucida is removed off, right? Zona pellucida is the one which is important for implantation at a right area, right? We also have studied about what is implantation, what are the abnormal sites of implantation, what is ectopic pregnancy during last class. Okay, last embryology section. So today what you have to understand is once the zona pellucida is removed off, what happens? Here we have got outer layer of cells. This is called as trophoblast. Now the trophoblast divides into two different layers. Now see here, you can see that there are two different layers. One is the cells which doesn't have any specific cell membrane. They're all, the nuclei are floating in and around. Okay, so this is a syncytium. Syncytium means what? It's a common area where so many cells are joined together. So this particular type of cells are called as syncytiotropoblast. And the other layer of cells which have got specific cell membranes, this layer of cells are called as cytotrophoblast. So we have syncytiotropoblast and cytotrophoblast. Okay, so there are two different layers of trophoblast. Now all of you clear till here, everyone? 8th day what happens? The trophoblast divides into two layers. What are the two layers? Syncytiotropoblast and cytotrophoblast. Clear, everyone?
Yes, ma'am.
Yes, ma'am.
Right. Now this syncytiotropoblast is very eroding in nature. Wherever it goes and sticks, it starts removing off the uterine endometrial cells, and it will start taking this entire blastocele, entire embryoblast totally into the endometrium. Okay, so it starts eating up the endometrium and pull the entire structure inside the endometrium. So see here, this is the syncytiotropoblast and cytotrophoblast. Okay, now what we are doing, we want to look into inside the cavity, so that's why we have taken a section of it and we are looking at the section. Please don't forget, this is like a complete sphere. Okay, it is a sphere, but we cannot see what is inside. So what did we do? We had taken a cut section of it and we are looking inside. So what is the next step happening? Syncytotrophoblast, cytotrophoblast, that is the first step. Next, what does happen? The embryoblast cells, which are at one corner, they will start separating out into two different layers. Some cells accumulate above, some cells accumulate down. Okay, these cells which are above, in the next picture you can see that, see here, some cells arrange in upper layer, and some cells arrange in the lower layer. These upper cells are called as epiblast. Epi means above, blast means early cells. Okay, epiblast. Now these are columnar cells. Okay, and the cells which are at the bottom, they will make a cuboidal appearance, and these particular cells are called as hypoblast. Okay, hypo means below. So what is the next step after syncytotrophoblast and cytotrophoblast? This embryoblast cells will divide into two different layers. What are the two different layers? Epiblast and hypoblast. Till here everyone clear? Guys,
Yes, ma'am.
Yes, ma'am.
right. Now see what is happening. We'll go back. This is the epiblast. This is the cytotrophoblast. Okay. In between the cytotrophoblast and epiblast, there is a small space developing. In between the cytotrophoblast, this is the cytotrophoblast, this is the syncytotrophoblast, this is the epiblast, okay, and cytotrophoblast. Between these two, there is a small layer of cells coming, okay, covering that and enclosing a cavity. Okay, so this layer of cells which are above is called as amniogenic layer. What is this? Called as amniogenic layer. And this cavity is called as amniotic cavity. Okay, what is it called as? Amniotic cavity. Now you can see typical columnar cell appearance of epiblast. So what is the next stage we will see? We will see after formation of epiblast and hypoblast. From the epiblast, there are a group of cells which will come above and separate the epiblast cells from the cytotrophoblast. Okay, so this layer of cells are called as amniogenic layer and the cavity is called as amniotic cavity. Till here clear?
Yes, ma'am.
Clear all of you?
Yes, ma'am.
Right. Now from the hypoblast, you see here, now slowly there is a small layer of cells coming down. Okay, you can see that these cell layers are coming down and they will start enclosing the entire cavity. Okay, 9th and 10th day, what happens? We will see a complete amniotic cavity. You can see that implantation is completed. Okay, entire the structure, embryonic structure is inside the uterine cavity. You can see a fibrin plug. This light green area is a cytotrophoblast, and outside more dark green area is called as syncytotrophoblast. Now what are the changes in the syncytotrophoblast? You will see that a spaces happening inside, that is called as, you can see that inside the syncytotrophoblast there are cavities developing here. What are these cavities are called as? These are called as lacunae. You can see this trophoblastic lacunae. Okay, what are these called as? Trophoblastic lacunae. Clear everyone?
Yes, ma'am.
Now next change, from hypoblast, there is another single layer of cells, okay, which are coming separating this blastocele cavity. Before it is called as blastocele. Now this layer comes out, okay, separates this cavity from the cytotrophoblast, just like amniotic cavity. Same like that, here also there is one layer coming out. What is the name of the layer? Name of the layer is important, name of the cavity is important. Here we say amniogenic layer and amniotic cavity. Here we say, what is this layer called as? Huesner's membrane, okay. HEU, see here it is labeled here, Huesner's membrane. Yes, NER, okay, it is Huesner's name, Huesner's, HEUSNER, okay, Huesner's membrane. Once this membrane comes, now this cavity is called as primitive yolk sac, okay, primitive yolk sac. So 9th and 10th day, what are the changes happening? Changes which are happening outside the embryonic cavity and changes which are happening inside the embryonic cavity. Outside, the changes are syncytiotropoblast grows well, cavities appears inside. These cavities are called as lacunae, trophoblastic lacunae. And inside the embryonic cavity, what are the changes? We get amniotic cavity and primary yolk sac. Okay, primary or primitive yolk sac. What are the layers covering? From hypoblast, we get Huesner's membrane. Okay, and from epiblast, we get amniogenic cell layer. Till here, everyone clear?
Yes, ma'am.
Yes, ma'am.
Right. Now 11th and 12th day, what happens? See the difference. See here, 11th and 12th day into the lacunae. What is happening into the lacunae? Can someone tell me what is happening inside the lacunae? What is happening here? Guys, answer. What is this?
Is it blood?
Yes, it is blood. Okay. Whose blood it is?
Mother's blood.
Yes, this is all endometrium. From the endometrium, maternal blood vessels will start erupting and pouring blood into the trophoblastic lacunae. So this is the first site of uteroplacental circulation. What is it called as? First site of uteroplacental circulation. First time maternal blood is entering into the embryonic tissue. Okay, this happens in the second week. Okay, all of you clear with this? What is happening here? This is the first sign of uteroplacental circulation. Second, what is this dark green colored area called as? I want answers fast. What is this dark green colored area?
Syncytotrophoblast.
Syncytotrophoblast. What is this light green color area called as?
Cytotrophoblast.
Cytotrophoblast. Right. Now see here what is happening between the cytotrophoblast and the embryonic cavity. You can see that there is a brown color area here. Okay. Brown colored area here. What is this brown colored area? We'll go back and we'll try to see this. See the picture? Syncytotrophoblast, cytotrophoblast and the embryonic cavity. Between the embryonic cavities and the cytotrophoblast, there is nothing. 9th and 12th, sorry, 11th and 12th day, you can see that there is a brown colored area. Lot of cells are present here. So these cells accumulate together and form a substance called as extra embryonic mesoderm. What is it called as? Extra embryonic, extra outside embryonic. Outside the embryonic cavity, we are getting mesoderm. What is it called as? Extra embryonic mesoderm. In extra embryonic mesoderm, what we will see? We will see a cavity appearing slowly. Okay, we can see that there is cavity appearing slowly. See here, now what will go in 11th and 13th days? Can you notice the difference? All the cavities in the extra embryonic mesoderm join together to enlarge. Okay, we'll go back. All the cavities in the extra embryonic mesoderm join together and enlarge. Enlarge in such a way that this extra embryonic membrane divides into two layers. One layer which is close to the cytotrophoblast and one layer which is winding around the embryonic cavity. Okay, we'll go back to this picture. Now see here, there is one layer which is outside and one layer which is covering this embryonic cavity. Okay, so this layer which is outside, it is called as somatopleuric. Somatic means peripheral. Okay, somatopleuric extra embryonic mesoderm. Okay, what is this brown color layer called as? Somatopleuric extra embryonic mesoderm. The layer which is covering the embryonic cavity, that is called as splanchnopluric extra embryonic mesoderm. Okay, what are these called as? Splanchnopluric extra embryonic mesoderm. There are two layers which are present, somatopleuric and splanchnopluric extra embryonic mesoderm. Are you guys clear? Okay, are you all clear with this? Any questions?
Yes, ma'am.
Okay, so what is this stage? In this particular stage, what is happening? See here, the changes which are happening outside, the changes which are happening inside. The changes which are happening outside are, you will see that completely lacunae are filled with maternal blood. Okay, you can see that the syncytotrophoblast is becoming very thin now. Okay, lacuna are enlarged. You can see the first appearance of this one placenta. Okay, placenta contains these cord-like substances which are called as villi. Okay, what are these called as? Villi. In placenta we will see villi. So this is the first site of appearance of placenta. Okay, how does it appear? It appears like a cord-like structures. What are these cords? These are the cords of syncytotrophoblast and cytotrophoblast. Okay, which are present in between the lacunae. Now lacuna are enlarging and cyto and syncytotrophoblast are becoming thinner and narrower. Please keep that in mind. Okay, so what you have to remember is appearance of primary villi takes place in this particular day. Second, inside the embryonic cavity, what are the changes? You will see that there are two layers of extra embryonic mesoderm, that is somatopleuric and splanchnopluric extra embryonic mesoderm. Because of this separation of these two layers and enlarging of extra embryonic cavity, this primary yolk sac becomes very small. We'll go back to the previous picture. We'll go back to the previous picture. See here, this is the primary yolk sac. It is large. Extraembryonic cavity is smaller, just forming. Now what happens in the next picture? This cavity is increasing and this cavity becomes smaller. Okay, now you see here, this cavity is increasing huge and this cavity becomes smaller. Okay, right? And whatever is extra is present, that is taken off like exocoelomic cyst. It is removed off, pinched off. So this yolk sac becomes smaller and narrower. So this smaller cavity is called as definitive yolk sac or secondary yolk sac. Okay, clear? Definitive yolk sac or secondary yolk sac. So this is the final change of the second week. Second week may now see how many cavities, what are the changes. It's a week of twos. Okay, what is week of twos? It has two second week. Okay, two layers of trophoblast, that is syncytotrophoblast, cytotrophoblast. Okay, two layers of extraembryonic mesoderm, that is somatopleuric and splanchnopluric extraembryonic mesoderm. It is having two cavities, amniotic cavity and definitive yolk sac. It is having two germ layers, that is epiblast and hypoblast. Okay, clear all of you? Guys, please answer. Are you all clear?
Yes, ma'am.
Yes, ma'am.
Now this is a point where we will try to observe what are the competencies. In competencies, the list, we will study what is pregnancy test at this point. Okay, what does pregnancy test? It is called as strip test. In this strip test, what does it do? Actually, the strip is had to be dipped in the urine sample, preferably early morning early morning urine sample of a female who is suspected to be pregnant. Okay, how suspect that whether the female is pregnant or not? How we can suspect? That is when the female misses her regular menstrual period. Now if you see, go back, which is the day 13th and 14th day post fertilization, means before fertilization. When does the fertilization happen? On the 14th day to 16th day of ovulation. So that means if a woman enters into a menstrual cycle, okay, her 14th day will be ovulation, her 14th to 16th day will be fertilization, and next 13th to 14th day, that means almost you are getting around 1 month period. Okay, so again she should be going back to her menstrual cycle. If that menstrual cycle is not happening or her menstrual periods are missed, so that time they can have a suspect, we can suspect that the female can be pregnant. So to test that, we have very sensitive pregnancy tests available. That is, first is, this is called as UPT, that is urine pregnancy test, which is usually over the counter available in the medical stores. So you can get that strip and check it, preferably in the early morning sample, urine sample. When it is dipped, you will see if it is positive, you will see there are two lines. Okay, if it is negative, you will see only one line. One line is for control and T is for test. If two lines are there, then it is positive test. Okay, so this is something called as urine pregnancy strip test. Okay, why do we get it? Because of the starting of secretion of HCG. What is HCG? What is HCG?
Human chorionic gonadotrophin.
Yes, human chorionic gonadotrophin hormone. Okay, so this HCG hormone is secreted by placenta. Okay, we saw that This is the first site of development of placenta from the syncytial trophoblast. Okay, so from here the HCG hormone is secreted. HCG hormone is also secreted from where? It is also secreted from the corpus luteum. Okay, now corpus luteum is called as corpus luteum of pregnancy because the fertilization and implantation has been done. So from here the HCG is secreted, which can be accumulated inside, not accumulated, can be entered into the maternal blood, and via blood it can be secreted into the urine, and it can be detected by the urine pregnancy test. Okay, so this is the secretion of HCG, that is the urine pregnancy test module. That is one of the applied feature of second week. Other than that, what else we can see? We can see sometimes at this level what happens, the pregnancy cannot develop. Okay, it might stop at this level, it may not go into the third week. Okay, so it at this particular time what happens, it might form form something called as molar pregnancy. Okay, this appears like uterine fibroids, the female starts bleeding heavily, and it cannot be stopped. So sometimes when we do a normal ultrasonography, we'll find that there is a molar pregnancy present inside the uterine cavity, which is not developed completely. So once you open that molar pregnancy, this is the entire uterus. Okay, you can see the uterine cavity, entire uterus has been removed. This is how a uterine cavity appears, and you can see that the molar pregnancy appeared something like this, which will lead to the continuous bleeding of the uterus. Other than that, we will also see something called as, which is very common, placental abnormalities. That means placenta, if it is present at one corner, okay, usually what is the site of implantation? Upper segment of the uterus. Okay, this is the upper, clinically upper segment of the uterus is called as the normal site of implantation. If sometimes if it is present in the middle segment, this is called as marginal placenta. Usually early pregnancy, if marginal placenta is present, it can slowly, as the uterus grows, it can slowly go upwards. So what usually these pregnant females are given, they say that yes, this pregnant female should be taken adequate bed rest so that the placenta may not erupt and cause bleeding and can cause spontaneous abortions. So these pregnant females are given rest. Sometimes what happens, if it is, see here, this is also called as low lying. That means lower segment of the uterus, if it is placenta is developed, then it is called as low lying placenta. Okay, this is also a dangerous, it can lead to spontaneous abortions. Now this is the most dangerous one for the fetus, when placenta is completely obstructing the internal os. Internal os means internal opening of the cavity. Okay, this is called as internal os, external os of the cervix. Now internal os is completely closed by the placenta. This is the most dangerous. This is called as complete placenta previa. Okay, so this condition, what happens is very common to have spontaneous abortions where any structures enters into the cervix. Remember that it will cause immediately constriction of the uterus, sorry, not constriction, contraction of the uterus, and can cause expulsion of the uterine contents. Anything which enters in between the cervix, it will immediately cause contraction of the entire uterus, leading to the expulsion of the fetus. Okay, so this is how complete placenta previa is seen. So types of miscarriages, not so important. This is important for medical students. You can just have a overview. It is missed, threatened, inevitable. So this is something, placenta previa. Okay, inevitable. So definitely it starts expelling. These are all types of miscarriages. Okay, this is incomplete miscarriage. That means only portion of the pregnancy has been released and some part of it is inside. Still it leads to continuous bleeding of the uterus till entire components of the pregnancy are being not expelled. So complete miscarriage is along with the fetus, the entire fetal material, okay, that entire thing should come out. Okay, if it is not coming out, it will start giving rise to the bleeding continuously, and it might cause lot of hemorrhage for the this one. Okay, so types of abortions not required for you. So this is just a video to see. There is a summary of the second week.
Implantation of
We'll
the
quickly see
blastocyst usually.
implantation onwards. We will see this is the blastocyst. Zona pellucida is gone. Syncytotrophoblast, cytotrophoblast. Okay. You can see the number of days here. Next, what happens? Syncytotrophoblast grows. Trophoblast, and syncytotrophoblast slowly enters inside the uterine cavity, uterine endometrium. Okay. Now see here, you can see lacuna appearance, bilaminar embryonic germ disc, epiblast and hypoblast. Okay. You can see the cavities formation. Amniotic cavity has been formed. Slowly from the hypoblast, Huesner's membrane will be developed, giving rise to primary yolk sac. You can see that now. This is the Huesner's membrane, and you can see primary yolk sac formed. Now you can see trophoblastic lacunae outside. So primary yolk sac, this is called as amniotic cavity, cytotrophoblast, syncytotrophoblast. Now the next layer is extra embryonic mesoderm, which is developing in between the embryonic cavity and the cytotrophoblast. At the same time, look at the syncytotrophoblast, how it is changing. It is growing, lacuna are developed. Now maternal blood will flow into it. Okay, you can see that on day 12, the changes you can see. Maternal blood flows into the lacuna, establishment of uteroplacental circulation. Now in between you will see lot of space. Okay, here extra embryonic mesoderm will develop, and inside extra embryonic mesoderm cavities will develop. That cavity is called as extra embryonic coelom. Now see, the cavity formation will make this primary yolk sac into smaller yolk sac. Okay. Now there are two layers here. This is the first layer, that is somatopleuric extraembryonic mesoderm. This is called as planktopleuric extraembryonic mesoderm. Extra part, whatever is not required, has been removed off, pinched off, and the smaller cavity is called as secondary yolk sac. Okay. Now see, extra embryonic coelom is so large, huge. Okay. And inside that cavity, this embryonic cavity is simply floating around with just one connecting area. So this is the future umbilical cord. Okay. So that connecting area, future umbilical cord, is called as connecting stalk. Okay. Which we will see in the third week in detail. Clear, everyone? Did you all understand this?
Yes, ma'am.
Yes, ma'am.
Right. So what are the questions expected from this second week? Yolk sac, extra embryonic mesoderm. Okay, these are short note questions which are expected from this second week. Okay. Any questions? No questions? Okay. So you can study till here for your examination. And anything you would like to ask, you can ask me. Sakshi, please send the attendance. Nothing is there, we
Yes,
will
ma'am.
our session here. Someone is asking, ma'am, we have to color the diagrams in theory paper. Yes, you'll have to color the diagrams in theory paper. Okay, colors, specific colors I already told you. Artery red, vein blue, nerve yellow, lymphatics green, muscle brown. Okay, these are standard colors. Other than this, rest all you can use anything. Okay. Okay then.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you, ma'am.